So, if-then statements are how you put together hypotheses and test them. So, if we say A, therefore B is true, then we can prove that hypothesis. Or the opposite, which we'll talk about. So in this case, we could say, if there's one morphology, that equals one species. Is that true? No. No, right? Because we have seen that when you can have the same morphology and be two different organisms, right? So it's not always going to be different. Another way you can look at it is backwards or upside down. Is if you have greater than one species, right, two or greater, you should have two or more morphologies. Sometimes that's true, right? But not always, as we saw. We had where they looked very similar, unless you really looked at it closely. You had two very similar morphologies and yet two different organisms. Right, so this hypothesis is not true, right? Because A and B are not always true. You don't always have one species in one morphology. So if either one of them don't match up, then our hypothesis is false. So in other words, a hypothesis disproves that the first part of the statement is true while the second part is false. Right? They got to match up. Right? Both sides have to match up. In neither one of these cases do we have this to be a truth. Right? That two species always equals two morphologies. Or that one morphology equals just a single species. Still with me? Right? And part of our proof to that was the plates that we looked at, right? Where we really kind of had one morphology on that one plate where they were both kind of white. Unless you really looked at them closely, then you'll see that we had a mixed culture. We knew we had two different organisms, and yet they don't look very different. So we cannot. If a hypothesis can't be refuted, then you have supporting evidence towards it. And some hypotheses that have been supported for very long times get called what? Anyone know? Theories. Good. Good. So, I'm trying to remember. This just puts it into, just reiterates. And so for an example, later on in the semester in Lab 11, we're going to look at the Piglo system, and we're going to look at fluorescence. And in fact, one of the organisms we have too, it produces a pigment that's fluorescent, Pseudomonas aeruginosa. And we found that out recently because Peter was reading one of my students' actual poster there, and he said, "Oh, it says it's fluorescent here, let's see." And we shined the UV light on it, and lo and behold, it fluoresced. I was like, oh, that's pretty cool, didn't know that before. I wonder if they could use that in clinical settings. If they probably not. The green is enough. With Pseudomonas, that green pigmentation, when someone has that kind of infection. Yeah. Oh, yeah. Yeah. Because actually the reason why my student did that poster, you see the picture of that model there? She got an infection with this organism, and it spread system-wide. And, I mean, she even lost her limbs, and it still wasn't enough to save her, and she ended up dying. And from a bladder infection with this organism. Right? So, pretty scary stuff. That's why she was saying, you know, if you see that in a clinical setting, that's a really bad situation. Right? Really bad situation. So, we use a scientific method, right? The first thing we do is form a hypothesis, right? Based on observations, right, or prior knowledge. So when you look at scientific papers, right, they're organized in the organization of the scientific method. You'll have an introduction, right, where they're going to introduce previous information that they've gathered, and they're going to form a hypothesis that they're then going to test. So they're going to say, if A is true, then B is true, right? And vice versa. If B is true, A is therefore true. So why, and also why are they doing experiments? Why are they asking this question that they may be asking? What's the significance behind it? And then methods is what they're going to actually do to test this hypothesis, right? So they're going to design an experiment or a series of experiments to test that hypothesis or hypotheses. More than one. The goal is to devise one or more experiments that will clearly disprove each hypothesis. Right. That seems kind of backwards, right? But if you can disprove something, then it's definitely not true, right? And so it's more of a process of elimination. So then when we have to go through and over the processes in your mind, write them down, so that you execute the protocols correctly. So that's why, right, for homework, you guys write out your protocols, right, so you're familiar with it. And then hopefully you follow it, right, so that you get the correct, you get accurate results. Not necessarily correct, but accurate. So no technical mistakes. So how do I perform these experiments properly? And then once you do the experiment, you're going to have results, right? Some tangible evidence as to what's going on. You'll make observations, you'll record them, you'll do the same thing. You'll record them sometimes in words, sometimes we take pictures, right, depending on the types of things we're doing. We may organize that information into graphs and figures and tables. And obviously you're not going to include stuff where you know you made a mistake, right? It's invalid, right? You would not include that. Unless you, of course, accidentally discovered something new. Then, of course, you'd report it. So in the case of Alexander Fleming and the good old mold, penicillium that produced penicillin, supposedly he discovered that by accident. So it's not what was not his intention. But, of course, he shared that information, right? That phenomenon. So what kind of outcomes should I record, and how should I report them, right? These are things that you think about, right, as you're doing these experiments. And then the last section is a discussion. So what do these results mean, right? Justify any ambiguous or contradictory results. Draw conclusions. What does this mean? What does this tell me? And a lot of times in the next section they're going to have further tests they want to do, right? Based on this information, this is where we could potentially go in the future. In other words, what does it mean, right? What do we know? So today, we're going to do 2 specialized stains, right? And we're going to get results. And based on those results, we can draw conclusions, okay? And so, again, we start out with the hypotheses, right? That they either have or don't have these structures, right? Or different components. So we'll look at each one of these using the scientific method. So peptidoglycans and glycoproteins are substances that you'll find in the cell walls of microorganisms. Peptidoglycan is unique to bacteria. Most bacteria have this substance making up their cell wall. It's made of alternating sugars, N-acetyl glucosamine. I always get messed up on these. And N-acetyl muric acid. And so they alternate, right? So you see you have the green one, the red one, right? In an alternating pattern. And remember, we're talking about 3-dimensional organisms. So this is completely coating and covering the cell. So almost think about layers of a jawbreaker, right? Most of you guys know that candy, right? Layers upon layers. Those layers are hooked together by amino acids. So 4 amino acids, right? 4 here. And then for gram-positives, because they'll have lots of layers of this peptidoglycan, they have an additional linkage, this cross-linkage, which is also an amino acid. Several amino acids linked together. So their name actually has to do, this name, peptidoglycan, has to do with the structural components of this molecule, the composition of it. Peptido refers to the bond between amino acids. That bond between amino acids is referred to as a peptide bond. Glycan is another name for sugar. So that represents the alternating sugars you see in the structure. And there is a particular linkage between these molecules, how the sugars bind with each other. And that's what they're talking about here when they say the 1,4. So the 1 and 4 carbons from each sugar next to each other are what are binding. And this is important because, for instance, did we talk about glucose? I get my not glucose, cellulose, which makes up the cell wall of plants. We didn't talk about it in this class. So that's glucose. But the way the glucose is hooked together, our body doesn't have the enzymes to break apart those sugars. So we can't digest cellulose. When you eat cellulose, when you eat plant matter, we refer to that as fiber. It goes one in, one out, in one end, out the other. And probably the best example of that is if you eat corn kernels. Corn kernels are basically embryos. It's a single cell of a corn plant. And if you don't break the cell wall with your teeth, it will go in one end and right out the other. Right? So when you poop corn kernels, it's because you haven't chewed your food. Chew your food, especially your plants. Right? You got to break those cell walls. If you don't have the enzymes to digest that cell wall, right? You can't break down cellulose. Cows can. But it actually is not them that's digesting it either. It's the microorganisms in their gut. Right? Bacteria that can digest that. So that's why linkages are important between cells, because you need specific enzymes to grab on and pull these things apart. Now, glycoproteins, so glyco is what, do you think? Sugar, and then, of course, proteins, which are made up of amino acids. So the protein components will form these long alpha helixes. And again, because they're structurally different, they'll interact with the dyes and the things that we do differently. So here's the sugar component, right? And here's the protein component. So when we look at bacteria, and when we stain them, we classify them based on how they come out in the gram stain. But that information correlates to what their cell wall structure probably looks like. Right? This is how they're affected with the dyes. So every cell always has a plasma membrane. Gram-positive cells will have a thick layer of peptidoglycan. In addition, here, these little squiggly things drawn in here is tectonic acid, or teichoic, acid. And it's almost like rebar, right? It helps stabilize the lot of layers in addition to that interbridge that I was talking about. So these guys could have up to 30 layers of that sugar hooked together by the amino acids. So in addition, the tectonic acids are there to help stabilize that very thick cell wall layer. Why do they have a cell wall? This helps the bacteria hold those specific shapes that we've looked at, and also helps them resist osmotic pressure. They're a single-celled organism, right? If water comes rushing in, what would happen to them if too much water comes in? They would burst, they would explode, right? Just like our cells, we don't have cell walls, so you put them in a hypertonic a solution, they're going to burst, right? They don't have the way to help protect them. These guys do have the cell wall, which keeps them from bursting under those types of conditions. Otherwise, they'd probably die all the time, right? If they are in that type of environment. In contrast, gram-negative bacteria, again, all bacteria, all cells are going to have a plasma membrane. If they have it, they'll have a relatively thin layer of peptidoglycan. So notice it's only showing a couple bars here. And then they tend to have another membrane called the outer membrane. And this is different from the plasma membrane or the normal cellular membrane that cells have. These have their outer leaflet, instead of being phospholipids, are lipopolysaccharides. So in addition to being a phosphate head and having those fatty acids, those lipids, they have additional pieces that stick up that are made out of sugar. Saccharide is another name for sugar, and of course poly means many sugars. So to give you an example, E. coli is a gram-negative bacteria. It has all of these levels. Its particular lipopolysaccharides can be used to differentiate even different strains of E. coli. So there are some pathogenic strains of E. coli out there, like E. coli 0157 you've probably heard. The O refers to these polysaccharides. The 157 is the number designation to that specific strain. Okay. The bad news with that one is highly pathogenic. Most of us have E. coli in our gut, right? But it's not pathogenic. If it was pathogenic, you wouldn't be in class right now. You'd be in the bathroom, right? Because it would be causing disease, right? And you'd be experiencing the effects of that disease. So because of these structural differences, they're going to be affected by the stains and decolorizer we use in the gram stain differently. And that's why you get the different results. And when we say positive and negative, we really are talking about a result in this gram stain reaction or test. Gram positive means that it stays stained with the primary stain, which is crystal violet, which is going to be a purple color. Negative means they don't retain the stain, the primary stain. They get stained by the counter stain which we use, which is safranin. So in the process, they're going to get decolorized. Right? We're going to remove the crystal violet from them, and then they're going to get stained again with the safranin, which is a red dye. But because in order to remove the crystal violet iodine, we're going to damage the cell membrane and wall of these cells, they won't retain a lot of that red dye, and so instead they're going to look pink. So as I said, 30 plus layers, or in this case, nanometers, really thick layer of peptidoglycan for the gram positive. So remember, the resolution of our light microscope is only about 200 nanometers. So we can't see, even though it's pretty thick, you can't see that cell wall. We'd have to use other methods like electron microscopy to see that. So I linked you guys up with this gram stain animation, which most of you guys in electric class have already gone through. So the first part goes over the actual staining procedure, which also I had posted. Looks pretty good, the professionally done one, right? The audiovisual department here at Delgado came in and videotaped me doing that, and put together and edited that video for me. I think they did a really good job. And so you can really see, and the reason why I wanted to do that one is you can really see one of the most important steps, and that's the decolorization step. All right, and that's where most people go awry. So today you're going to start with an already heat-fixed emulsion that I'm going to give you, and then you're going to stain it. So you're not going to have to wait for it to dry. You're not going to have to heat fix it. You can just go right to gram staining. And it's going to be a mixed culture. Some of you guys are going to get a mixed culture slide where it's going to look something like the picture here. Where is it? There it is. Well, you'll see purple cells and pink cells together. Others will get a slide that have them separated in two little circles. So one circle is going to end up being purple, the other one's going to end up being pink, right? 1 gram positive, 1 gram negative cell. So they tell you different times here But in microbiology, when you're doing stains like this one, the 1 minute rule, right? If you do it for about a minute, you're good, okay? So we'll do crystal violet first, right? A minute. Rinse with water. If you were to look at it in a microscope, they'd all be purple at that point. Then you're going to add the iodine. I think in your guys' lab manual, unfortunately, it says grams reagent or something like that. It's iodine. So make sure that you fix your procedure if you wrote it from your book, right? That grams, whatever they say, is actually iodine. Right, what step would it be? Gram stain. They say gram stain. It's actually iodine. This isn't going to change the color of the cells. Instead, something else is going to happen, right? We're going to end up creating a large complex of the crystal violet and the iodine. We're going to make it a larger molecule. So you're going to rinse with water after a minute. Again, as I said, they'll still be purple. This next part I will demo for you guys too, especially if you didn't watch my video, so you can see it live. That you want to do this, you're going to do it over a paper towel so you can see what you're doing. You want to do it drop-wise, and you want to do this until the dark purple drops turn light purple. You don't want to do it until they're clear. You'll decolorize too much, and even your gram-positives will have the dye rinsed out of them, and you don't want that to happen. Then you're going to rinse with water, and then the last stain is what? Safranin for a minute. Rinse, blot dry, and then you're ready to look at it under the microscope. Notice if you were to stop after the decolorization step, you would only see your gram positives. Your gram negatives would be colorless, right? You wouldn't see them, but we want to be able to see them, right? So you would counter stain with your safranin, and so they would come out pink. Why do they come out differently? It's because of the different structural setups of their cell walls. So gram positives, right? All cells have their membrane, and then they're going to have this thick layer of peptidoglycan. This layer is affected by the ethanol that we use to decolorize. So the first step, crystal violet's going to go into your cells. Because remember, it's a basic dye, so what charge? Positive, and it's attracted to those negative components of the membrane, right, and of inside the cell, your DNA. So it's going to go inside that cell. Then the iodine is going to go in. And they're actually going to combine together to form a larger molecule. So then when you do the next step, which is decolorize with ethanol, what happens is that this layer, this peptidoglycan layer, it has big holes in it, right? It's just structurally sound, rigid. It doesn't stop stuff from going in and out, okay? Just provides support. So that stuff comes in, combines, forms that larger molecule. The ethanol, the effect it has on those sugars is it shrinks them up. So those holes between the molecules get smaller. So that essentially traps in the crystal violet iodine. Also, that complex is not very soluble, right? So it kind of crystallizes inside those cells. So it's harder to get it out. Gram negatives in comparison, these guys, crystal violet, iodine's going to go in, but because they have such a thin layer peptidoglycan, and they usually are going to have that outer membrane is dissolved. Big holes between the two membranes will happen because ethanol dissolves lipids, right? Membranes are mostly made out of lipids. This is true, too, if you use hand sanitizers, right? They dry out your hands. Because guess what? It's ethanol. It's alcohol. It was probably isopropyl. It's still alcohol. And what is it doing to the lipids? Dissolving them and removing them, right? And that's what keeps your hands nice and moist, right? And soft. So that's why they're trying to add, moisturizers to those things nowadays so they don't dry your hands out so much. So you'll keep using their products. Right. So the problem here is, right, they're not going to be trapped in anymore. The crystal violet iodine is going to get washed out. They're going to be clear when we look at them under the microscope. So usually to help you guys visualize, this is one of my favorite toys, right, from good old kiddo days. This toy, as you see, has two parts, and it has a handle at the top. It actually comes apart. The little blocks just barely fit in the holes that match the shape of the block. So little kids, the first time they play with this, they stick the blocks all in, and then of course they want to do it again. So what do? They don't know it opens. They stick their little hand in one of those holes, right, and then grab one of the blocks. And then they try to take their hand out. Can they get it out? No, because the combination of the hand and that block are too big, right? Can't get it out the hole. So then you have to convince the little one who's crying to let go of the block, take their little hand out by itself, and show them how to open it, right? So the same principle is as with the crystal violet iodine, right? Crystal violet goes in, iodine goes in, but once they're in, they combine. And then even worse, the holes what? Shrink. So imagine if the little kid stuck his hand in there and the hole shrunk. They'd really be crying, right? Really would be stuck. It would be scary. So a little intelligence quiz, those you guys in my lecture can't answer. How do you get that ball out the top? What could you do? What's preventing this ball from getting out that top hole? These little pegs? Could you maybe take them out? That's probably how they got it in, right? You take the pegs out, you take the ball out, right? You can put the ball back in without the pegs in it and then add the pegs, right? And you're definitely not going to get it out. I like Peter's example better. We'll get to it in a minute. So, again, we looked at the structure of crystal violet, right? It becomes this positively charged. It loses its chloride ion. Chlorine is very similar ionically. If you look on the periodic table, it's in the same column, right? So here's chlorine, here's iodine. The difference is iodine is bigger. It's a bigger molecule. So when we do the switch out, right, with the chlorine and the iodine, and it bonds better to the crystal violet, again, we get that larger molecule, right? Size-wise, it ends up being 123% bigger, the iodine, than the chlorine molecules. So this is Peter's stray cat who's adopted him. As you can see, she can barely fit through the fence to get into his yard. He feeds her, she has to stay, because guess what? She can't squeeze through after she eats. Just that little difference, right, of eating a meal, she can't make it back through the fence. So she usually stays for a while. So what do they call her? Can't say her name. So the same thing here, right? It makes it much harder to get the molecule out because it ends up being bigger. With that addition of the iodine. And as I said, it's less soluble, right? So it doesn't go into solution well and stays in the cells.
So like we use ethanol or alcohol or whatever, and you shrink it. Does it shrink it to its normal size since it gets bigger with the iodine?
No. Think about this. This is what I'm talking about. Crystal violet. Good question. Thank you. So you're getting things confused is what's happening. So crystal violet comes in. Crystal violet is actually a complex with chlorine. When the iodine comes in, it switches out the chlorine for the iodine. It binds with the crystal violet and creates that complex. The difference here is that the chlorine is a smaller element than iodine. So in addition to now instead of being crystal violet and chlorine, it's crystal violet and iodine. And the iodine is larger. So it makes the crystal violet larger when it complexes with the iodine. Okay, so then the next thing that happens, right, is when we decolorize, we shrink the hole. So it makes it even harder for that larger molecule complex, the crystal violet iodine, to get out. Make sense? Cool. All right. So if we were to look at what happens in each step, right, in detail, for gram-positive cells, when we stain them with crystal violet, right, it's a basic dye. It's going to go inside the cells, and it's actually going to go into all cells, but we're just going to focus on the gram-positive right now. They're all going to be purple. What happens when we add the iodine? Goes in, right? Binds to the crystal violet. Makes it a larger molecule now. It's a crystal violet iodine complex. So much longer, it's complex. Now when we decolorize with ethanol, do we actually decolorize, remove color from gram-positives? No. We shrink the cell wall and we trap in the crystal violet iodine. So these guys retain the dye complex. They stay purple. Next step, staining with safranin. Well, these guys are already purple, right? When we stain them red, are they going to change color? No, they basically become a darker purple. And probably the best way to understand dyeing is laundry, right? So I did this once when I was a kid. I accidentally put a red sock in with the whites. My dad had pink underwear for like a week. He was none too happy with it, but it was underwear. Hey, no one sees it, right? Underneath. Okay? So it bled, right? It bled onto the whites. Now, had I say, if my dad had black underwear, which is one of the reasons why I love my husband, yeah, black. If I throw a red sock in and it bleeds, are we going to notice? No, because the black is a much darker color, right? When you wash darks, you might have bleeding, but you're not usually going to notice it, right? Because it's not going to affect the color enough that you notice it. So what happens here is, even though you stain it with red, because purple is much more vibrant color on the spectrum, it just ends up being a darker version of the purple, right? It still appears purple. Still with me? All right. So gram-negatives. First step, crystal violet, right? What color of the cell is going to be? Purple. Second step, iodine. What's going to happen? Yeah, it's going to make that complex, right? Crystal violet and iodine are going to bind with each other. It is this step where we become different, right? This decolorization step. Do we decolorize these cells, gram-negatives? Yes, right? And again, what effect does it have? It's going to dissolve that membranes, right? It reduces the ability of the outer membrane to trap in the crystal violet iodine. Again, too thin of a peptidoglycan layer, if there is even one there. It's not enough to trap it in, right? The cells are going to become decolorized. And at this point, if you were to stop, you would not see them. They'd be clear or colorless. So we've got to counterstain. When we do that, this is a red dye, goes into the cells. But are they going to be able to keep a lot of red dye in? No, right? So they're going to be lightly stained like my poor dad's underwear. They will be pink. So in comparison, right, this is the very important step, is the decolorization step. You mess up this step, right, you're not going to get accurate results. So, drawn out graphically, again, first step, both of them, whether they're gram-positive or gram-negative, are going to get stained purple. We're going to add the iodine. It's going to create a complex. If you look under the microscope, they still look purple. This is referred to as a mordant sometimes because it adheres, right, and makes it thicker, makes it larger. The next step is the decolorization, right? This is where we get the differences. In the video, they talk about porosity, right? So how well something comes out through the pores. So it decreases porosity because we're shrinking those holes, right? and it gets trapped in for the gram-positive. For the gram-negatives, we're increasing porosity, right? We're creating lots of pores that the complex can come out. So they become clear. And then the last step, so these guys still have the crystal violet iodine. These guys don't have it anymore. So therefore they're clear. Last step, we're going to counter stain. Both of them are going to get stained. So again, notice a darker purple and a light red or pink for the gram-negatives. Oh, what if you make mistakes? Right? So as it says, we're going to look at each mistake individually and how it would affect our results and why. So let's review. What are our 4 major steps? Crystal violet. What's next? Iodine. Next. Ethanol decolorization. Counterstain with safranin. So, what color will gram-positive cells be, purple or pink or colorless, if you fail to add the iodine? So let's go over each step. Well, the first step is crystal violet. You do that one, all the cells are going to be what? Purple. You skip the iodine, you forget. So we're going to get a larger molecule. So what's going to happen when we decolorize? We might actually decolorize these guys, right? The crystal violet might get washed out and make them colorless. So then we're going to counterstain with safranin. So what color are they going to end up being? Pink. Is this a correct result for gram-positive? No, they're supposed to be purple. So the crystal violet step is the crystal violet and then iodine step in that order is very crucial, right, to getting accurate results. Because the gram-negatives, if we skip that step, right, so they're going to be purple. Forget to add the iodine. When we decolorize, are they going to get decolorized? Yeah, like normal, they still will. And then you're going to stain with safranin. So they'll be pink. So is this the normal result? Yeah. Except what color are also our gram-positives because we forgot the iodine? Pink. So can you tell one from the other? Okay. What if you forget to decolorize? So your gram-positive cells are going to be purple, right? You're going to add your iodine. They're going to stay purple. We're going to oh, nope, we forgot to decolorize, right? So, forgot this step. So they remain purple. Then we're going to stain with safranin. But remember, they're still going to look purple. So for gram-positives, forgetting that step, not a big deal, right? Because they were going to stay purple anyways. But what about our gram-negatives? They're going to end up being purple, right? Because you're going to stain them purple. You're going to add your iodine. If you don't decolorize, they're going to stay purple. Even if you stain them with the safranin, they're going to be purple. Is this what they're supposed to be? No. Right? So, you forget decolorization, you're going to end up with 2 purple cells. You can't tell positive from negative. Take-home message is every single step is important, right? Done in the correct order. Okay, so next one, safranin. So gram-positives, is this going to affect them if we forget that step? Slightly. They're going to be purple, but just not as dark purple as they normally would be, right? So again, we're going to stain them purple. We're going to add the iodine. They're still going to be purple. We're going to decolorize. They're still going to be purple. We don't stain them with safranin. Oh, well, they're still going to be purple, just not a dark purple. It'll be a lighter shade. But yes, right, since the shade of purple looks about right. And trust me, shades are really hard to determine, especially when you're not comparing one to another. You wouldn't really notice. It's when you get to your gram-negatives, if you did a control slide like you should, crystal violet, they're going to be purple, right? Iodine complex, they're still going to be purple. You're going to decolorize, they're going to be clear or colorless. If you forget the safranin, are you going to see them? No, they're going to be what? Colorless. So, of course, is this a correct result? No. Because we're not even going to see them. The last one's my favorite, and I actually did this just to see. What if you reversed the order? What if we made safranin our primary stain that we did first, and crystal violet the counter stain? So for gram-positives, right, we're going to stain them what first? Real safranin. In this case, they're going to be red. Right? So we're going to stain them with a safranin dye, which is red. They're going to be red. We're going to add the iodine. Apparently it does complex with safranin as well. When you decolorize, it retains that dye, so they stay red. When you stain with crystal violet, they're now going to become purple, a dark purple. Of course, this is a correct result for gram-positives, right? It's when we get to the gram-negatives it's going to create some problems. Because first we're going to stain them what color? Red. Crystal violet complex. When we decolorize, they're going to be clear. But now we're going to stain them with crystal violet, so they're going to be purple. Oops. Is this a normal result? No. But I can tell you because, again, as we said before, right, when you stain them with both dyes, the crystal violet and safranin, it's going to be a darker purple than just purple alone. So I've done this one, and you do actually end up with darker purple gram-positives and lighter purple gram-negatives.
So
Yeah.
the difference?
It's noticeable, but trust me, you don't want to try and you don't want to do it that way. Because if you don't have a comparison, right, I had both on the same slide. If you don't have a comparison, how are you going to know if it's light purple or dark purple you're looking at? It'd be like impossible mission. It's much easier purple and pink, right? So that's why the order of the dyes, too, is very important in those contrasting colors. So both of these dyes, safranin and crystal violet, are basic dyes, right? We can use them to do simple stains. But in simple stains, we're just using just one dye, right?
Mm-hmm.
So can you determine gram-positive and gram-negative in that case? No. What is it about the differential stain that creates these differences? What's that important step that creates the differences in the gram stain procedure? Decolorization. Right? Oh, those are my hint questions. The ability of gram-positive to retain the crystal violet when subject to alcohol decolorization. And of course, this is enhanced by the use of the iodine. So, if you saw large eukaryotic cells in a preparation from your gum line, like if you were to take a scraping, you'd probably pick up your cells, your cells, your epithelial cells, your squamous epithelial cells from your mouth, as well as the bacteria that are present in most people's mouths. Trust me, everyone's got them. If we were to do a gram stain of this, what would your cells, your eukaryotic cells, end up being? Gram-positive or gram-negative? Negative. You know from lecture. Okay, so let's talk about why they would be negative. So here's a picture of them. And those normal people, this is kind of a joke, everybody would come out negative. They would come out pink. And the reasoning for that is, think about this. Crystal violet was going to be the first stain, then iodine. When we decolorize, what is it about bacteria that keeps the gram-positive positive, keeps that crystal violet iodine in? The cell wall. And what's the cell wall made out of? Peptidoglycan. Do our cells have cell walls? No. And they're certainly, therefore, not made of peptidoglycan. Now, there are other microorganisms you could stain, and they're going to either be positive or negative based on whether the crystal violet iodine is retained by those cells. But for our cells, it doesn't happen. It does not get retained inside the cells. So we decolorize and they end up becoming pink. Another clue here in this picture, this is this very large cell. What's this structure within the cell? It clues you in that it's a eukaryote. It's a nucleus. These purple things are all gram-positive bacteria. You still have a question?
Yeah, it's kind of like, I just want to know, like, if you did a plant, I know they have cell walls, and I know they don't have peptidoglycan, but like,
Right.
you know, would they be able to, like,
It depends on how. So our question is, what about if you stained a plant cell with the gram stain? They have a cell wall, but their cell wall is made out of cellulose. I have to say I've never experimented with that. It would depend on whether that cellulose would actually be able to trap in the crystal violet iodine, if they would come out positive or gram negative. I'm not even sure the dye would even penetrate cellulose. I don't even know if you would be able to stain them with these dyes.
Do they already have a pigment? Or
Yeah, and a lot of them already have their own pigmentation, so most of the time people wouldn't even bother staining plant cells because you don't really need to. Although they do stain them in some preparations, like in biology lab, when you guys have looked at the roots, they do special stains to be able to see the xylem, which is the section where the water runs through in the cells and stuff. So sometimes they do stain them, but they're using specialized stains that I'm not aware of. Good question, though. All right, so when we think about the gram stain and the scientific method, right? So why am I doing this experiment? Right? What do we do? What's the method? How do we perform it properly? This is our procedure, right? What outcomes, what results can I report? And so in this case, what are you going to see?
the differences in the cell walls.
Yeah, but can you really see the differences in the cell walls? You can infer that. What are you actually going to see? It's not that hard, y'all. What colors?
Oh,
Pink or purple. That's what you're going to see, right? But then that's going to draw conclusions, right? What does that mean, right? So in the case of purple, what does that mean? Thick peptidoglycan cell wall. Case of negative, not a thick peptidoglycan cell wall, right? And probably the presence of that outer membrane. So we want to identify the fundamental structure of the bacterial cell wall with gram stain. That's the question we're asking. How do we go about that? Right? We're going to gram stain. Right? We're going to follow this procedure in the correct order for the correct time frame. Because otherwise we won't get accurate results. And so what are we going to see? Right? Gram positive are going to be dark purple. And gram negative are going to be pink. How will we record this? We can record this in words, right? We could sketch drawings. We could even take photographs. What does it mean, though? Right? And as we said, gram positive means you probably have that thick peptidoglycan layer, right? And it's very rigid. About 40% of the known bacteria out there are gram positive. About 60% are gram negative. So it's not quite half and half. And these, of course, don't retain the crystal violet iodine, right? So they have a very thin, if not any layer at all. So it's just as we talked about with our cells, right? We don't even have peptidoglycan or cell wall, right? And so then they come out pink. Although most bacteria are going to have that structure of membrane, thin layer peptidoglycan, and an outer membrane. So that brings us to our next thing that we're going to look at, and that is glycocalcics. And we're going to be looking at a particular type referred to as the capsule. So cells that have these, and not all cells do. So here's our cell. Here's the cell membrane or cell wall, depending on what cells you're talking about, right? And it would be the outermost layer labeled 6 here. So glycocalcics refers to sugar coat. So most of them are made out of sugars. A sweet husk. So a network of polysaccharides that project from the cellular surfaces outside the bacterial cell wall, right, and membrane. Occasionally they contain proteins or glycoproteins, and there is at least one case where it's exclusively amino acids. It's a little bit different. And it can be a capsule layer or a slime layer. The difference is the capsule layer is pretty uniform all the way around the cell. It's quite firmly attached. The slime layer is not uniform and is not firmly attached. And some organisms actually use this for motility, right? They kind of slide along the slime that they produce. We're interested in the regular ones known as the capsule, where they're going to have this uniform structure. Why is this a good structure for bacteria to have? And in fact, we're even going to look at a eukaryote today that also has this capsule structure. They aid in attachment to surfaces. This is particularly important for pathogenic organisms, to be able to stick. And for ones that get into hosts that have immune systems like ourselves, it helps them hide, evade our immune system, evade a very important process called phagocytosis that we're going to take a look at, where our white blood cells literally will eat, gobble up and eat bacteria. But when they have a capsule, we have a hard time recognizing them. It's like they're wearing a disguise. So to do this staining procedure, it's going to look very much like the negative stain you guys did previously. So we're going to end up with these red slides, like I have on my light box right now, because I've done the first half of that staining procedure. So remember, this is going to stain what? The background, right? It's a negative, it's acidic dye, it's going to stain the background. So we're going to add the drop. We're going to add in our bacteria, we're going to use a spreader slide, and we're going to spread it out. We're going to completely air dry. Do not heat fix these slides. Right? Leave it on your light box until it's dry. Do not let anybody look at a red slide. Because if you look at a red slide and put oil on it, we cannot finish staining it. Okay, the next step is going to be adding the Maneval stain, and I'm going to do that in a moment to the ones that I have. Because this contains acetic acid, which is the same thing as vinegar, so it'll have a vinegary smell to it, which some of us don't like me. My students this morning, they're like, "Ooh, what's that smell?" I'm like, it's the reagent. We're going to leave that on for 1 minute, then you'll rinse it off, and then you'll blot dry it, and that slide is going to change from the red color to kind of a grayish color, right? And that's when you know you're done. Right? So as I said, if anybody tries to focus a red slide today, stop them, right? Because I didn't finish staining it. This is going to stain the background, but it's also going to stain the cell, right? And you guys stained this organism before using just Congo Red, and sometimes we saw a little bit of a cell on the inside. This time you should definitely clearly see the cell on the inside. And there should be a halo around it because the capsule does not stain. And remember, oil immersion from now on, right? Always with bacteria, you got to go all the way to oil. So it should be a clear white halo. So again, it's like a negative stain. And then we're going to kind of simple stain with that Maneval stain. So in your book, it says to add a drop of serum before adding your bacteria. We skip that step because we find we don't necessarily need it. But why do they say it could be useful? It'll help adhere the bacteria to the slide. The ones with capsules sometimes are kind of slippery. We haven't had any problems with them not sticking. So we skip that step. Saves us money. Saves us a step. Serum is the component of, it's a liquid component of your blood after your blood's allowed to clot. And it's really the proteins in that liquid that help stuff stick. So oral bacteria produce a capsule. And again, this is related to these functionalities that I told you about. So why is this beneficial for them to have a capsule, the ones that you would find associated with your teeth, for instance? To help them stick, right? And again, as far as our immune system is concerned, It's going to help them hide, right? It also protects them against one of the components of our immune system, which is antibodies, for a certain amount of time. It takes us a while to recognize them, produce antibodies that are effective. So this is hopefully similar to what yours will look like, where again, if it has a capsule, you'll see the bacteria in a clear halo around it and the background will be stained. Except ours is going to be more of a gray background than this picture here. You should see the shape of your cell. This one's pretty much a rod shape, as you can see, right? Or bacillus. There's one of the organisms you're going to stain today that does not have a capsule. And it should look something like a picture I have coming up. So why do we have to do these two different stains, these two different dyes? Well, think about this. If we just did, which we did last time, right, just the Congo Red, negatively charged dyes just stain the background, right? If this is the only step, then you're not going to necessarily see that capsule. So, for instance, Micrococcus luteus, right, we just saw a round, clear space when you negative stained it, right? So, as I said, do not stop here. Right? It's only the first part. Why using the basic Maneval's reagent? Remember, think about charges. So this is going to be positively charged. This is going to stain the cell. But it's also going to be attracted to the dye that you already put on the slide. So that's why the whole slide is going to change color. Right? And there's actually a pH change that happens, which is a major region for the color change. So, is the capsule stained by the dyes? No. It's not charged, right? And so therefore it does not get stained. So it appears as a clear halo around your cells if they have it. So one of the organisms you'll look at today, which is one of the cocci, notice here that it's purple and then the grayish background. You see no clear halo around these cells. This one does not have a capsule. I'm not going to tell you which cocci it is. You guys got to figure that out today. Or see if you can. It's tough. They're tiny. So, what if you did a simple stain? Would you be able to see a capsule? No, because even though I have a line drawn here, it would just stain the cell. And where's the capsule located? Outside the cell. And it's going to be clear, and it's going to blend in with the clear background, right? So definitely no way you're going to see. Now, negative staining, again, most of the time you're probably not going to see the capsule because the cell itself won't get stained, just the background. So in this case, it might appear a little bit larger if you were to compare a simple stain to a negative stain, right? But you're still not going to be able to visualize that capsule. You got to do both steps. So, again, why would we do this? We want to identify if your cell has this capsule, right? Helps us in identifying, especially if we don't know what the organism is, which in the second half of the semester you're going to ask this specific question. How do we do this? Well, we do the capsule stain procedure, right? Don't forget stain that red slide. Okay, what outcomes are we going to have? What are we going to be looking for? What type of results? Right, so we're going to be looking for that clear halo around the cell, between the cell and the background. And these will vary in size depending on the organisms too. Some of them are really thick, some it's very thin. Negative, no capsules. You saw in that picture, right? No halo, right? Just going to be purple cell, grayish background. How do we record, right? We're going to make drawings. We're going to write it down in words. Maybe take photographs. What does this tell us about the organisms? Well, if they have a capsule, right, we know that they're probably really good at attaching to surfaces, right? And if it's a pathogen, it probably helps them evade our immune system. So, for instance, Klebsiella pneumonia, which is one of the ones you're working with today, that's why it's so effective at causing pneumonia. It can stick to us, and it can invade our immune system and cause quite a bit of damage to the lungs, right, before we're able to defend ourselves. Another virulent bacteria that you find in your mouth is Streptococcus mutans, right, and this is the one that deteriorates your teeth when you eat too much sugar, right? When you feed it sugar, it produces acids that deteriorate your teeth. Again, it's encapsulated so it can stick to your teeth, right, attached to that surface. Gram-negative means they lack that external protective layer, right? So they're probably not as effective at attaching and are more vulnerable to phagocytosis, more likely to be eaten. So I'm not going to read through the glossary of terms. They're in the PowerPoint for you guys as reference. The one thing I do want to show you is that on Blackboard, there's a folder that says videos. And I have the actual videos down here where you can like download them to your computer if you want. But I also have linked them up with YouTube. I'm just going to show you this first one that's showing one of our white blood cells chasing down and engulfing by phagocytosis the actual bacteria. These are other ones about how important it is that our cells have this ability, right, to help defend us. Those of you guys in my lecture class, we'll look at these later when we talk about immunology. But they're my favorite videos, and if you're interested in the immune system and how that works, those are there for you to check out. So I'm going to mute this so I'm not listening to her talking. So they literally just took blood from a graduate student, put it on a microscope slide. You see red blood cells. This is a neutrophil. It's a specialized white blood cell that does phagocytosis. They added some staphylococci bacteria, so notice there were cocci in a cluster. These are not motile, but it's in a liquid medium and they're kind of bounced around by thermal energy, like the normal colliding between molecules. This guy produces chemicals the bacteria does, just in general. Just like, like when we sweat, we produce that smell. And so that chemicals he's chasing after, the neutrophil is chasing after him. And then he engulfed it, right? So you see how he grabbed it up, right, and engulfed it. And that neutrophil almost looked like a protozoa that we've looked at, right? Kind of moves like what? Like an amoeba, right? So they can push their cytoplasm, right, to move around. They move in that manner. They don't have flagella or cilia or anything like that. They have an amoeboid type of movement. This is just whatever the next one that comes up. All right, so last